very much through my colleagues in the industry is every sample is a human, a patient, a loved one. My friends, your friends, people you know will have probably been tested using things that I've done and my team has done and my organization has done to help them navigate their own healthcare journey. And the proximity, even as a tube, every tube is a person asking a question. They're fearful. They are, am I okay? We do no harm. Beneficence. We do our best for that patient. And it gets even deeper than that.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello, and welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin, a biotech science enthusiast. Along with me from North Carolina is my excellent co-host, Dr. Chad Briscoe, a biotech scientific expert. And we publish BioTalk Unzipped twice a month and bring experts from different corners of the pharmaceutical, biotech, or med device worlds to share their innovations, insights, career journeys, and personal stories to the show. On this episode, episode 7, we are bringing in Dr. Russell Grant, VP of Research and Development at LabCorp, to the stage.
Dr. Russell Grant earned a PhD in chromatographic and mass spectrometric technologies from the University of Swansea, Wales, United Kingdom. He continued his scientific training in various industrial settings, which have included senior scientist at GSK, principal scientist at Cohesive Technologies, technical director at Eli Lilly, and director of mass spectrometry at Esoterics Endocrinology. When Esoterics was acquired by Covance and later LabCorp, Russ's impact grew over the years as he rose to his current role of VP of Research and Development at LabCorp. Russ has pioneered the use of direct injection technologies, chromatographic systems multiplexing, utility of automation, and new analytical platforms for application and bioanalytical applications. His research goals are focused upon improvements in speed, sensitivity, and quality of liquid chromatography with tandem mass spectrometric analytical systems and assays. Leveraging his knowledge of mass spectrometry and passion for perfection, he has been at the forefront of implementing mass spectrometry and clinical analysis. Dr. Grant serves as Clinical Chemistry Chair for American Society of Mass Spectrometry and is a member of the American Association for Clinical Chemistry. In addition to his exceptional career contributions within his companies, Russ is also a gifted science communicator. He's a highly sought-after speaker at regional and international meetings where attendees come for both leading-edge science and strong opinions delivered in an entertaining manner. I think today's discussion will certainly be evidence of that.
Wow, that's a setup. Thank you, Chad.
Welcome, Russ. How are you?
I am good, gentlemen and viewers. How are you, gentlemen?
Doing
Very
great,
good.
Thanks.
Yeah, looking forward to diving into it.
Yeah, I might just clarify a few things. So Esoteric
bet.
Endocrinology was acquired by LabCorp, which also acquired Covance. So the sequence there was a little different.
Yeah, okay.
But yeah, jumped into diagnostics 20 years ago after about 8 years in pharma. So.
And that was actually one of the things I wanted to explore with you, Russ, and it was maybe down the list a little bit since we popped into it there. Talk to me a little bit about that, because I think I first came to know you back, I don't know, 99 or 2000. Mm-hmm. There was, one of the ASMS meetings. Mm-hmm. You were still in bioanalytics there. Mm-hmm. Moved into Cohesive. I knew you a little bit there. And then at some point, you made that jump into diagnostics. Tell us about that a little bit, because I've never really heard that story from you.
Yeah. So, it goes a ways back, but as a child, I was just enamored with American television, cop shows and things like that. And so I always wanted to flee the nest and go as far and as high as possible, which made me realize a couple of things. I needed to do something that was unique and put myself in positions where I was uncomfortable to grasp those opportunities. I worked in pharma for two great pharmaceutical companies, SmithKline and Lilly. As a journey, SmithKline in the UK, Eli Lilly in Toronto, but I was always coming to America, and Canada was a wonderful hybrid society for my wife at the time, still my wife, thankfully, and I. And I actually did 16 job interviews in the United States, and I got 16 job offers. Now, I don't think I was that good. I think I was just perhaps a little cheap. But either way, 15 in pharma biotech, and then this one in clinical diagnostics at Esoterics. And it was in Thousand Oaks, which was just down the road from Amgen. I also got an opportunity with Amgen, but I thought, you know, 95% of every drug I ever worked on, well, that time and effort was on a drug that didn't make it. And I worked on a few that made it, so kind of rare. I thought maybe let's pivot that equation and just see if clinical diagnostics was the right fit for me to do the right fit for me to do something interesting. And so I made that jump, and frankly, I haven't looked back. I'm almost zealous in my energy and desire to use the tools, not just mass spec, but flow and immunoassay and automation to drive these sort of high-end analytical workflows for patient care. I'm just zealous about it. So it was taking a jump, knowing that there was a security blanket because I'd done well in the interview process, but really trying to
Sure.
figure out, do I want to spend my time in a world where methyl ethyl propyl futile was the iteration in chemistry, and I mean no disrespect, they were great companies to work for.
Sure.
But now in pharma, 95% of every project or program or workflow that I've ever put my time and my team's energy towards makes it to clinical care. So it's a complete pivot for energy. But yeah, I was always coming to America, and I just wanted a massive roll of the dice after working in pharma for a while, Chad.
So Russ, we try to balance between getting too deep in the science here and having it accessible to others. But one of the areas that you really educated me on, and I would say a few others, but is that difference between bioanalytical for clinical trials and applications of mass spec and bioanalytical for clinical trials versus in clinical and healthcare and the applications you've been working on with esoterics and with LabCorp over the years. Can you just, in a nutshell, kind of sum of that up for us?
Oof, that's a good
I
one.
know. Yeah.
Um, we grew up with the iterations of the bioanalytical method validation guidance,
Mm-hmm.
the Crystal City
Mm-hmm.
meetings, and that put some pretty good quality guardrails around how we would develop assays for exogenous therapeutics, things like that. When I stepped over into clinical diagnostics 22 years ago now, it was all about endogenous analysis, which was a whole new sort of problematic
Mm-hmm.
field, generation of calibrators, spike and recovery studies when it's an analyte that's on board. So the actual technical challenges to create externally calibrated, internally standardized assays were initially following the GLP, bioanalytical method validation guidance. But pretty quickly, I learned that there is a whole slew of additional workflows following the Clinical Lab Standards Institute or CLSI guidance workflows for any clinical applied test, if you like, for direct patient care. So I got deeper into when was a mass spectrometer telling me the truth and telling me lies for endogenous analyte measures, and a whole slew of different statistical ways of thinking about error as a total error, a component of bias and imprecision. And then what is the distribution in normal and then abnormal and the separation of those states. So it really just deconvolved what I thought mass spectrometry was fit for, what we could use it for, and what the guardrails were of accuracy and imprecision, as we know from GLP guidance, back into that may not be good enough, or it may be too narrow based on the distribution of biomarkers in a disease in a healthy state. But that's a gradation. So it was a good starting point. Certainly, the documentation standard and the accuracy standard
Mm-hmm.
as a foundation to start from was a great place from drug development, BMV. But moving into diagnostics, you have to take a lot more into consideration. Probably the most challenging is that we are not controlling the population. In fact, quite the reverse. It's an anybody, anytime, any state of health, and the markers that we I started certainly working on, have interferences, concomitant drugs, metabolites, new drugs on markets, creating new metabolites to create new selectivity issues. So the assays were never finished, and selectivity was the biggest challenge, I think.
Yeah, and I think one of the things you pointed out to me once, you were talking about an assay that was really challenging, and I can't remember the assay, but I think you told me you were working to save 7 seconds per injection, and in the clinical, in the bioanalytical for clinical trials, of 7 seconds, big deal. That's just the difference in a needle wash or something like that. We're doing 1,000 samples; it doesn't really add up that much. But it really it struck me because of the numbers. Tell me about the numbers of assays you're doing. Whereas a laboratory like the one I work at, we're talking a few thousand samples, per study.
Yeah, so probably a good idea for me to reflect on the biggest clinical study I ever did was I ran the phase 2, phase 3 clinical
Hmm.
studies at SmithKline, and that was around about 45,000
Hmm.
specimens. Massive study,
Massive.
massive. We have assays that run on one or two mass spectrometers here that do more than 10 times that in the same time frame. So the clinical study ran for 18 months, although the analytics was about 10, 11 months of that. We,
Yeah.
we, we are 10 times that number on LCMS here per year. And that assay has remained unchanged for 12 years. Multiple sites running multiple instruments, but literally 400,000 samples for that assay a year. And a lot of the pressure is the turnaround time. So trying to turn that data around back to the clinician and the treating physician as fast as possible, 24, 48 hours from draw. A little challenging when we have sites scattered through the United States and ex-US shipping samples in. But yeah, the scale is 10x easily. And when you think about that, and then the longevity of these assays that we have to support, we have to really think about robustness, and all seconds matter because that's scalability, that's multiple instrumentation platforms to support that volume, which again continues to grow, particularly post COVID as people are stepping back into their healthcare journey, and we're starting to see people be a little bit sicker, unfortunately, from taking a little bit, maybe a hiatus from the wellness, or even the
Yeah.
the lingering effects, of course, of COVID. So the scale is about 10x, and the longevity of these assays can be 10 or 20 years. Amazing, just so different. And I remember the assay. It was the assay was testosterone.
Okay. Yeah, I'm sure it was. Yep.
Hmm.
Yep. I had a feeling you'd be like Chad. I remember that conversation. So,
Hmm.
you segued actually into something I want to ask you about as well. So thanks for that. COVID. I don't really know what role you might have played in COVID, but I'm thinking you're at LabCorp. I think it's well known that LabCorp certainly played an absolutely pivotal role in COVID and detection. But what is a mass spectrometrist leading that research? What was your involvement, and what did you guys do to answer the tough questions?
Not
What
mass
were those tough
spectrometry. Well,
questions?
first of all, not mass spectrometry because it's simply not fast enough.
Hmm.
So a couple of things I would like to just comment. I think across the continuum of health and care, right the way from physicians and nurses and point of contact and collection, right the way through to even pharmaceutical companies opening up their research to each other and trying to solve for an awful time. I think as an industry, it is our Mount Everest. I hope we don't have another one in my lifetime. It was really challenging. LabCorp was fortunate enough to step forward along with many other major laboratories. We had the first FDA EUA approved assay for self collection using PCR. So not mass spec,
Hmm.
PCR, and some really elegant automated workflows. I actually worked on the serology side, so I was working on microsampling for anti antibody or antigen a little bit, but mostly antibody because we thought maybe antibodies could
Hmm.
mean something. Of course, the virus mutated, so the antibodies sort of changed over time. Went through a couple of publications I think you'll see out there using microsampling to track native versus immunological response from vaccination. Uh, so my part was certainly more on the serology side. We opened up the people from the team to support uncrating boxes. I mean, at our highest volume, we didn't have enough people to actually open boxes because every sample
Wow.
was its own unique container.
Yeah. Yeah.
I
Yeah.
think we're on record of somewhere in the region of 350 to 400,000 samples a day.
God. Wow.
And so in the
Yeah.
facility I'm in, we were actually hiring day labor to literally open boxes, and our leadership
Wow.
came through, and they would open boxes, so all the lab professionals who could run the analytical workflows were seconded there. It was It was a very, very
Hmm.
tough but an incredible time for the
Yeah.
industry as a
Yeah.
whole to really pull together.
Hmm.
But
Mm.
it was one of the most challenging environments and times I think I've ever seen. Again, LabCorp and other big diagnostic companies and the startups we saw really did step up to the mark there, and we did an
Hmm.
incredible thing as a community, and one of those proud moments that I don't want to do again.
Yeah. Yeah.
Right.
Well put.
Very good. Well, Chad, we ready to get into our new news segment?
Let's do it.
New news.
New
New
news.
news. So, did you know, and Russ, this is actually a former company of yours, that according to Fierce Biotech, that reported on January 23rd that Eli Lilly successfully restored hearing in an 11-year-old child who had been deaf from birth. And this test was specific to people with mutations to the autopharylin gene, and so it's hearing loss by any other means that cell gene therapy wouldn't necessarily help. But this is an amazing result, restoring hearing to an 11-year-old kid who has never heard anything before. Have you heard about this, and what do you think?
Personally, no, but I'm certainly going to check that out. And I hope one of the first things that he heard was the intro solo from Shine On You Crazy Diamond by David Gilmour, because that to me is
Right.
perfect tonality. But I will certainly be checking on that. Amazing.
Yeah, and we'll post links. We'll post links
Mhm.
below. Below the podcast and YouTube. I'm sure Greg will put that up there with the story. There was a story on Fierce, but it is phenomenal. It's what we do in this industry. You talked about in COVID, how we all came together and did an amazing thing, and then you think about the years and the shoulders of giant upon giant that we as an industry had to stand on to have
Yeah,
something like that accomplished, right? and
yeah.
and it's just phenomenal. I can't even imagine what a child who's hearing. I mean, what is
Right.
that to hear
I,
for the first time? So
I wish I
yeah.
could have seen, and I don't, maybe they videotaped it, maybe they didn't, they didn't want it to be public, whatever. But could you imagine the look on that child's face, hearing something for the very first time had to be just unreal.
Oh, yes. I mean, just to the overwhelming sensation that one could imagine. I mean, you can't imagine. We literally cannot imagine that.
Mm-hmm.
I've certainly done immersions in pools where everything's sort of closed off, a bit like, Tommy from the movie. Everything is black and you can't hear anything except, the dull thud of your heart. But even
Mm-hmm.
that's noise. So yeah,
Mm-hmm.
that would be phenomenal. I'll be checking on that.
Yeah. Yeah.
Yeah. And I think that's the promise of cell and gene therapy, the things that we can do that we never thought possible. So it's pretty exciting. And now, Russ, after you earned your PhD in Wales in
Hmm.
chromatographic and mass spec technologies,
Hmm.
did you fancy yourself moving to America? Was that always part of the plan?
Yeah, from before I went to college. In fact, when I was 19, I think it was, I was playing university college soccer football, the real one.
Hmm.
And somebody gave me Craig Whitehouse's paper which showed electrospray of proteins and peptides. So I kind of knew, oh, it's going to be
Hmm.
mass spectrometry. John Bynan and then Di Games had gone to Swansea. So I went from Cardiff to Swansea and I'm like, okay, I'm going to do that. I broke virtually every instrument I touched for the first 6 months. I learned how to fix every instrument I touched for the next 6 months. And I actually had a very soul searching moment. I was practically quite incompetent, technically not very strong. I immersed myself in the literature for a good 6 months. I'm like, well, I'm going to learn all about the theory of how these things work. I can kind of fix them. I can certainly break them. Um, and then try and really work on the application of extraction, separation, and mass spectrometry, and just try to combine those things together. So that was what I did. I realized that I think at the time that mass spectrometry was on the up, so that would be a rocket for me to get across the pond. I jumped onto the SciX3 Plus. I saw that particular instrument as being, potentially a legendary, and it did turn out to be a legendary
Hmm.
instrument in bioanalysis. So I saw the writing on the wall for the right mass spectrometer to work on. I was working on a different platform at college. Um, got into GLP, got into CFR Part 11, got into regulated bioanalysis, made the transition to Canada. So I took a few risks, but educated risks, I think, on that stepwise journey of what am I going to be good at and what are going to be enablers for me on that journey. But make the biggest the biggest sort of roll of the dice was to leave my entire family except my new wife behind and walk across and well, fly across the pond and just see how high could I go. I actually did an undergrad year, my third year at 3M in London, to ask myself the question: do I go out of undergrad or do I get letters? And I saw very, very quickly if you didn't have letters, the glass ceiling would squish me down. And I was the first one from both sides of my family to go to university. I didn't want a glass ceiling, so I realized I needed a PhD. I realized at Swansea I had access to lots of technologies to get breadth and some depth. I realized a Sykes 3 Plus would be an enabler for me. I realized drug development was evolving through the use of the Sykes 3 Plus. I realized I needed to become au fait with those approaches. I worked for two wonderful companies in SmithKline and Eli Lilly, and then jumped out of the industry for a bit into diagnostics. And frankly, I can't go back. I love this industry. I love what we do.
And you mentioned that earlier, you became zealous for diagnostics.
Yeah.
What made you zealous? What keeps you zealous for diagnostics?
I have had the privilege and the pleasure of interacting with patients whose diagnosis has been enabled by my work. Many, particularly when I was at Esoterics, working with pediatricians and pediatric endocrinologists and meeting families, mothers, fathers, siblings, the patients themselves that I've certainly developed assays to support clinical diagnosis. And when you meet the first person that you've had a positive impact on, it changes you. When you meet the 10th, it still changes you. When you meet the 50th, it's just you. I went home, I think, a couple of times and just sat out on my porch with a small glass of wine, just looking up at the stars and just thanking my guardian angel that there is a place in the world that I've been managed. I've had the opportunity to sit in and take my grab bag of skills and make a positive impact. And it's that immediacy, I think, and that proximity to the impact of your work. That really, really changed me. And you're not trying to reduce the timeline for a drug to get to NDA or IND. You're literally engaging back and forth with tricky clinical situations with the profession.
Every result's an immediate impact. I mean, that's what I'm hearing, right? So what? Yeah, yeah, and then meeting people that it helps and knowing that it's going to make an impact. I know I had a similar experience when I first started working in flow cytometry, and I'm by far no expert in flow, but I remember we were running samples for a late phase trial, and the drug was at that point treating someone, and the scientist in the lab said, "Hey, Chad, you want to understand flow better." He said, "Look at this." He said, "This drug is to shift the cell population. If it shifts it from here to here in this, nice scatter plot that you see, that means that the drug is working on this person." And he flipped through and he said, "Look, it's working. It's working. It's working." He got to like sample 6 and he said, "Didn't work with this person." Oh, wow. And you have no idea who it is, in that case, but it's carried with me that experience that, wow, that drug, that last line of defense didn't work for that person. But, looking at the bright side, it did work for a number of other people, and the drug went on. It was, I believe it was approved and it was quite successful, but
Certainly
it was
a
very
mantra.
sobering.
Yeah, exactly. I think a mantra that we use, that I hear very much through my colleagues in the industry, is every sample is a human, a patient, a loved one.
Mm-hmm.
My friends, your friends, people will have probably been tested using things that I've done and my team has done and my organization has done to help them navigate their own healthcare journey. And the proximity, even as a tube, every tube is a person asking a question. They're fearful. They are, am I okay? We do no harm. Beneficence. We do our best for that patient. And it gets even deeper than that. I mean, we are I work for LabCorp, one of the biggest diagnostic companies, I think the biggest on the planet, and Quest Diagnostics, another wonderful big company. I've picked up the phone and talked to experts at Quest Diagnostics to get consultants and guidance and some enablement as a community, because we actually, it sounds, again, very zealous, but the patient first mantra and mindset runs deep, and once you've been touched by that, it's the probably the only compass point that one ever needs in their career to make sure that you stay focused on doing the right thing by the patient. So when you say zealous, I can't think of any other way than doing the right thing for another human being. Many you'll never know, millions I'll never meet, but knowing that I've done my best within the confines of a wonderful company and enabling the patient to get the best outcome that we can provide them. And that kind of journey is one that I would pay every penny I've ever had to have again.
That's awesome. And it is so important. These diagnostics can literally be life-saving. I was reading in one of your publications, providing absolute certainty without absolute quantity, and you're testing the I believe it's kidneys for certain kidney defects, and you could do it by just a blood sample, which is pretty remarkable. And that way, people who may be receiving a kidney are receiving a kidney that is going to extend their life and not actually, potentially kill them. So that is, I mean, that's remarkable and really, really important.
That's a very interesting one. So that one is for ApoL1. And intriguingly, we have the genome that we know, and we have certain variants that lead to potential concerns around kidney transplantation. The gene expresses for the protein with different SNPs in the or different amino acid arrangements. We take that protein, we chop it up into small peptide pieces, infer the gene. Take that even further, they bleed on a blood spot card. We take that and we run that by mass spectrometry. So we're using protein fragments from digestion to define the protein to infer the gene.
Fascinating.
The capability inside diagnostic medicine and the speed of evolution is astounding. It's astounding.
I just noticed that I'm sort of getting chills, and I was sort of readjusting myself. What a nerd, because I'm just like I'm taking it in, and it's amazing. It gives you chills, right?
The things that we're working on now with again in neurology and oncology and CAR-T and things like that. The speed of science it's a little overwhelming, but actually with a really broad toolkit, which is certainly what I've tried to build in myself and my team members, we can get plugged into some fascinating technical challenges. And as solid analytical scientists and analytical chemists, we bring a lot to the table in terms of reduction of bias and improvements in imprecision and workflow. It's a wonderful hybrid for us to just access these radically changing ideas and concepts and be able to be plugged in at any phase. It's great. Have I sold it? Have I sold you on it yet, guys?
No,
yes.
I was sold.
Sign me up.
15 years ago when we had these conversations. Okay, Russ, I want to. One of the things we do is I ask an unzipped question. Right. This is one where this is one where I want to get a little controversial, and so you're ripping my heart out one way, and what I'm going to ask you rips my heart out in another way. But having known you and had the pleasure of so many talks and so many conversations, you're always quoting literature, this guy and that guy and this paper, and you check this out, and then send me the paper. And one of the things that I have been following lately is it's generally academic fraud, but it's really about fraud in publications and scientific publications, and there's that sort of balanced against the recent I don't know, wave against science in the news and things like that, and it rips my heart out. And so I want you to unzip this, because you're a guy who relies so heavily on literature. What can we do about this as scientists, and how are you dealing with it?
So I was thinking about this question. Somebody was asking me about why I do so much repetition, why I do imprecision and repetition and multiple instrument comparators and multiple people and add those variances. There's a funny quote. I don't even know who to attribute it to. A mass spectrometrist does an experiment once, a biologist will do it twice, and an engineer will repeat until they give you the mean free time to failure. And I have an engineering background, so for me the repeatability of experimentation is really key, and not overselling the initial hypothesis and the initial conclusions. Ian Wilson, who was somebody who worked for me during my PhD, he was an external professor that I lent heavily on, Dr. Wilson, taught me that a good scientist tries to disprove their first conclusion. A great scientist tries to disprove it with a different methodology, and a world-class scientist is always trying to disprove their conclusions. I think we take the CSI effect of a quick and easy answer from a mass spectrometer that's off the screen that they're going to squirt something into and it's given a magic result, and we present perhaps science as being a magic black box, and it isn't. I think the scientific method, as given to us by the methodologies of Roger Bacon and Isaac Newton, are tried and tested. We need to couch the conclusions we make with appropriate skepticism, but we're running against a media engine that is been overdosed with CSI and these crime shows. For me, reproducibility and repeatability in multiple settings in the multiple laboratories are key to taking a hypothesis to a rule to a law, the scientific laws that we honor. And it's the hypothesis testing and repeatability that is the gap that we see. Again, the scope and speed of science doesn't help us, as there's so many people doing so many things. There's interesting conversations going on right now in lipid analysis space, trying to define when is a peak and not noise, i.e. have I detected something versus not, because those have not been repeated. I'm aware of that going off in the literature, and I'm in lipidomics discussions, societies around when is a peak actually a peak and not manipulated noise. Repeatability is not sexy. There's no funding for repeatability or reproducibility. There's funding for hypothesis generation, and which is the Roger Bacon mode to open up new ideas of hypothesis and thinking. But there's a limited funding vehicle for reproducibility and repeatability. To me, that's a fundamental funding gap to remove the noise and really amplify the signal or get into repeatability. One of the things that I realized in my PhD, when I had my sort of moments of thinking, what is the right way to move ahead in my career, is to prove that I could repeat. And in diagnostic medicine, we have multiple instruments, multiple assays, multiple facilities. We do round robin comparators, monthly and 6 monthly for most of the tests that are available to us. And if we can't do a round robin with an external body providing specimens for those ring trials or round robin trials, we work with other diagnostic companies to switch calibrators and switch samples to make sure we at least agree with each other. So there's a lot of alignment in the diagnostic industry, just by design under CLIA. But it's the repeatability, and the funding's not there to repeat there, Chad. It's not the sexy stuff that gets the top 8-10% funding dollars paid out.
Thank you for listening to the BioTalk Unzipped podcast. This concludes episode 7 and part 1 of our conversation with the wonderful Dr. Russell Grant. Tune in on March 15th for episode 8 and part 2 of our conversation with Russ. Until next time, be well.